how you guys doing today? Today I'm going to be talking about Crohn's disease and the role of gut microbiota. My name is Cannon Metcalf. So, first thing we're going to ask is what is Crohn's disease? Crohn's disease is a type of inflammatory bowel disease that causes chronic inflammation anywhere along the gastrointestinal tract. Patients often experience symptoms like abdominal pain, persistent diarrhea, fatigue, and weight loss. The exact cause of Crohn's disease is not fully understood, but is believed to be driven by a combination of genetic predispositions, immune system overreaction, and environmental triggers, especially changes in the gut microbiome. The complex interplay is what makes Crohn's disease challenging to treat, and why there's a growing interest in how how microbiome might be influencing inflammation and immune responses in the gut. So, gut microbiome, friend or foe? The gut microbiome usually acts as a helpful partner, digesting food, making vitamins, supporting our immune system. But under certain conditions like stress and antibiotic use or genetics, the microbiome balance can shift. This is called dysbiosis, shown right here. When that happens, even normally harmless bacteria can become harmful. Some start to produce inflammatory molecules or even break down the protective mucus barrier in the gut. In people with Crohn's disease, this shift from friend to foe may be what drives chronic inflammation. And that's where bacteria like Rhamnococcus gannavis comes into play. So, my case study is mostly talking about a microbe called Rhamnococcus gannavis. And it has gained a lot of attention in Crohn's disease because of its effect that it may have. This particular bacterium is found in higher numbers with people with Crohn's disease, especially during flare-ups. What's important is not just present, but what it does. R. gannavis produces certain substances that interact with the gut lining and the immune system. These substances can trigger an inflammatory response, essentially agitating the immune system. Over time, this can lead to chronic inflammation, which characterizes Crohn's. What makes this microbe very interesting is that it doesn't just thrive in the inflamed environment, but it might actually help create it. So it's not just a passenger, but maybe potentially playing an active role in driving the disease. So our key question here is, could R. gannavis be actively contributing to inflammation by stimulating immune cells? And during this paper, they're going to try and answer this question. So the first thing they tested is, to begin answering this question, researchers asked whether R. gannavis produces something that directly activates immune cells, especially dendritic cells. So they cultured R. gannavis in a defined medium and then collected the spent medium. And this is the spent medium here. Essentially, this is a broth that bacteria has been growing in, especially our R. gannavis bacterium specifically. Then they exposed bone marrow derived dendritic cells to this spent medium and measured TNF alpha levels. TNF alpha is a essential cytokine during inflammatory responses that are secreted, if you don't remember. What we're looking at here is this, obviously the TNF alpha levels is showing how much secretion is going on under different responses, under different conditions. The control groups, the DMSO and the plain media, have very low TNF alpha levels, meaning that there's almost no background activation during this, obviously. And then LPS is a strong immune activator, which is shown as a positive control, so it shows that high levels of TNF alpha secretion are being shown. Interestingly, R. gannavis is actually being shown to be having a robust TNF alpha production, nearly almost as strong as LPS over here. But what they don't know is what R. gannavis is actually secreting. So they don't know if it's a protein or a lipid or any of those things. So what they did is they looked at specific key findings. They treat it with heat, proteinase, DNase, RNase, and even lysosomes. And when they tested with all this, it showed that obviously TNF alpha production was still very high. So what this tells us is that that the molecule that's being secreted is most likely a polysaccharide. Polysaccharide is the only one that's actually going to be able to withstand all of these tests. So this experiment gives us a first major clue. Argonavus secretes something that can directly activate dendritic cells and drive inflammation, and it's likely a polysaccharide. Okay, interactive question 1. What is the purpose of treating the media with proteinase, nuclease, lysosome, and heat treatment? I'll give you guys a second. Okay, and the answer is to identify whether the inflammatory molecule is a protein, nucleic acid, peptide glycan, or heat label compound. So this is kind of helping us point down, like specify what the molecule is looking for, what the molecule actually is. Because we know proteinase breaks down proteins, DNase RNase breaks down DNA and RNA, lysosomes can break down peptide glycan, and boiling it, getting it 100 can denature these proteins and specific lipids, showing that it is neither of those. Okay, so the next thing we're going to talk about is how does the immune system recognize this specific glucohormone, which is actually the polysaccharide that we identified. So far, we've seen that Argonavus secretes a heat-stable molecule, a glucohormone polysaccharide that strongly activates dendritic cells and induces TNF-alpha production, even more potently. But that raises the next key question: how does the immune system recognize this molecule? Our immune cells detect microbes using pattern recognition receptors, or PRRs. One major group of PRRs is toll-like receptors, TLRs, which is part of the innate immune system. TLR2 usually senses lipopeptides, while TLR4 is known for recognizing LPS, and in some cases, certain microbial polysaccharides. So, the researchers next asked, is this inflammatory response to glucohormone being triggered through TLR2, TLR4, or possibly another pathway altogether? That leads us to the next figure. So, to test which receptors required, they used dendritic cells from 3 types of mice. Wild type, right here, TLR2 knockout, and TLR4 knockout mice. So TLR2 knockout mice means that these mice do not have the TLR2 receptor, and TLR4 obviously means they do not have the TLR4 receptor. And then they did it stimulated with 4 different treatments. They did it with DMSO, they did it with LPS, PAM3-CIS, and our experiment, Argonavus glucohormone polysaccharide. So, obviously, when we look at DMSO, it shows that this is basically negative control. Nothing is responding and showing TNF-alpha production. LPS is very known for activation of TLR4 receptors. So, LPS uses TLR4 receptors to create a TNF-alpha response. So, when TLR4 is knocked out, there's actually very low TNF-alpha production, showing that it needs the receptor to produce it. When PAM3-CIS is shown, it is also known for responding to, it only binds to TLR2 receptor. So when the TLR2 receptor is knocked out, almost no secretion of these TNF-alpha cytokines are being brought about. And so when we look at our final graph of our experiment, Argonavus, it shows that when TLR2 knockout mice were exposed to Argonavus polysaccharide, it shows that there's still a very high level of TNF-alpha production. But when they're exposed to TLR4 knockout mice, almost no production of TNF-alpha levels are being shown. So what this tells us is that when TLR4 is being knocked out, there's no secretion, which says that this specific polysaccharide is binding to a TLR4 receptor. So when the receptor is not there, no secretion happens, no cytokines, and no inflammation. What do you think could happen if we knocked out the TLR4 receptor in the mouse model of Crohn's? Is it A, more inflammation, B, less inflammation, or C, no change? Correct answer is B, less inflammation. So, since we know that TLR4 receptor binds to the specific polysaccharide that we know of, If the receptor is no longer there, then what is the polysaccharide going to bind to? Nothing. So if the dendritic cell is not going to respond to it, no secretion of cytokines, no inflammation, so be. So here's kind of an overlook of our microbe right here. And then it secretes our specific polysaccharide, which then binds to the TLR4 receptor on these dendritic cells, which then creates a cascade of events to secrete cytokines, creating an inflamed gut, known as Crohn's. Okay, our third question. Which of the following best explains how Argonavus can contribute during... Which of the following best explains how Argonavus may contribute to inflammation during Crohn's disease flares? Okay, give you a second. It is C. It secretes a heat-stable polysaccharide that activates dendritic cells through TLR4. Okay, thank you guys so much. Great day.